Okay, so today's lecture and the next lecture are going to focus on the prefrontal cortices. Okay, so we're going to talk about cognitive control and executive function in both of these lectures. Now, so this is just kind of basically a comparison of the extent of prefrontal cortex in 6 species as indicated by the shading. So everything that you see in blue, that is a prefrontal cortex. Okay, now notice how small the region it is for the cat, dog, and squirrel monkeys. It's greatly enlarged in humans. Okay, and then you can see that the chimpanzee is definitely more when it comes to like other species of monkeys. So that's when you kind of think that the chimps are closer to human also evolutionarily compared to other kinds of non-human primates. Okay. Now, here are some real life examples. So, for instance, if you are overriding a habitual responses, so for instance, every time you get to a certain place, and let's say every time you go home, you are very used to park at a certain place, and then when you get to this place, and then you would kind of, let's say, go through the elevator, and then you go home. But today, you have to go to the store instead of directly going home. But somehow when you get to the elevator and then you're like, oh my God, how did that all of a sudden got home? I was supposed to go to the store. So this is you not overriding your habitual responses when it comes to the external cues. So which means that when it comes to the goal-oriented behavior, there are a lot of cognitive control that are involved. Okay. And another thing is distraction of inhibition of the distracting stimuli. So the things that you kind of have to do is everything that you are supposed to do, you are supposed to block out what is not important and focus on the prize. So in this, so to speak. Okay. And another aspect is solving new problems. Okay. And then the last one we're going to talk a lot on is shifting between different tasks. Okay, and this is really also the concept of multitasking when things are involved. Okay, so this means that there are certain aspects of the cognitive flexibility that you need to do in which that you have to constantly shift your attention from one set of rules to another set of rules while and at the same time kind of um, in this case in the switching task case, you have to kind of really keep an eye on what is exactly what you need to do at this point. Now, this is some of the cognitive tasks that involves conflict. Now, for instance, in the Stroop test, the written word might match the color of the ink that it's printed in congruent trials, and it might be different from it in congruent trial, or it could be non-color item in the case of like the neutral trials. And the congruent trials and versus the incongruent trial, of course, you have to, because the way that we perceive things are going to be much more automatic when it comes to the processing of words. So in the incongruent trials, your task is to name the color of the ink that it is printed in. So in congruent trials, you don't have much of this problem, but in congruent trials, you really have to inhibit your natural inclination to really say out the word green because your task that's asked of you is to tell the researcher what to tell the doctor the color of the ink that it's printed in. Okay. Now in the flank task, the central stimulus and the flanking stimuli might face the same, in congruent trial, we're opposing in incongruent directions. Okay, this is also has to do with whether or not if it's congruent or incongruent. Okay, now in Simon test, a cue appearing on the left or right instructs the subject to respond toward one of the response directions. The cue might the cue may appear on the required response side in congruent trials or on the opposite side, in which case that would mean that it's an incongruent trial. Okay, now, so A is kind of showing you 3 example tasks. Now in B, what they're showing you is these are the response latencies in congruent versus incongruent trial depending on the conflict level in the previous trial. Okay, now the graph basically show you the performance of human subjects in face name test. Okay, so face name test is basically can you match the person with the face that you of famous people that you know. So in this test, subjects see photograph of famous faces on which are superimposed the names of famous people. Now the subjects have to discriminate between politicians and actors based on either the photograph or the name. Okay, and the face and word might both belong to an actor or a politician, so that would mean it's a congruent trial, and or it might be differ in referring to actors and politician, and that would mean it's an incongruent trial. So that would mean high conflict. So that's what the H stands for. Okay, so when the face of the relevant dimension. So basically, when the phase was the relevant dimension, the reaction time in incongruent conditions were higher than in congruent conditions. What you see here. Okay. Now, however, the reaction time in incongruent conditions significantly decreased if the previous trial was also incongruent condition. What does that mean? That means the ones that are previous, basically the ones that was previous, that the trial that were previous to the target trial really has a huge issue when it comes to affecting the stimulus time. Okay, so which means that when there are mixed message and in which that you have to keep constantly changing incongruence to the congruence, or the congruence to the incongruence, and what the congruent to incongruent, which is not much of a problem that you see in the LL. But when you have when you have congruent followed by incongruent, in which what you see over here in this trial, then that is a major issue in which that you can see that the reaction time really suffered. Now in humans and macaque monkeys, the anterior cingulate cortex, so I'm going to call it ACC, and sometimes I will also call it anterior cingulate. This area is situated below the superior frontal gyrus. Okay, so this is the part that you are seeing left here. So everything that is circled. Okay, so that's the anterior cingulate, and so this is situated below the superior frontal gyrus and in the medial frontal cortex. All right, now the clues to the function of the ACC can be derived from its connectivity patterns. Okay, which include dense projections to the motor cortex and spinal cord, implicating that the anterior cingulate in motor control, and the projections to the anterior cingulate from the midline thalamus and brainstem nuclei, kind of also suggesting that there is an important role in arousal and drive states, basically kind of influencing the anterior cingulate function. Okay, so that would mean that the ACC is also implicated in modulating bodily arousal states. So how excited you are, okay? And so arousal state is not necessarily sexual arousal, it's just pretty much like a generalized arousal, whether or not if you are in a vigilant state. Okay, so for instance, when you feel that there is some sort of imminent danger that is coming to you, let's say when a person is following you home, and then you feel that you have a heightened vigilance about your surroundings, your ACC is really playing a major role in that. Okay, now the ACC also have a very strong reciprocal connections between the dorsal ACC and the lateral prefrontal cortex, the dorsolateral prefrontal cortex. Okay, so that would mean that the anterior cingulate plays a very important interactions in between these two areas in cognition. Okay, now when it comes to the actual dorsolateral prefrontal lobe, you'll see that this area it plays a very important role when it comes to executive function. So we might talk about it later in this lecture. Okay, now the more ventral aspect of anterior cingulate is well connected to the ventral striatum. Okay, now so basically like the orbital frontal cortex, the ventral striatum, and the medial temporal lobe are all connected to the anterior cingulate cortex. So this idea that this dorsal cognitive and ventral emotional ACC regions are also kind of proved kind of popular. So they kind of thought that the dorsal aspect of the anterior cingulate plays a more important role in cognition as opposed to the ventral ACC is more takes more role in the emotional aspect of the anterior cingulate cortex. Okay, now as you can see in the figure, the dorsal and ventral cytoarchitectural subdivision of the anterior cingulate cortex can be discerned in macaque monkeys on top. So top is the monkey brain, and the human is the bottom brain. Okay, so this kind of broadly corresponds to the anterior cingulate sulcus. Okay, so that is the delineate. That's the basic thing that delineates the superior frontal cortices and versus the anterior cortices. Okay, and so basically that is the part. Now, although there are some species differences that kind of exist, for example, there is a greater proportion of the area 32 that is situated above the what do you call this? Above the corpus callosum. Okay, so supracallosally. So in humans than in macaques. So that means that there is a greater area 32 in humans than in macaques. There are also similarities of the ACC that is going on in both macaque, monkey versus humans. So you can note that the rostral aspect of the anterior cingulate cortex, which is very close to the motor area, this is located in area 24. Okay, that you see. So the recent lesion studies in macaque monkeys, they have kind of determined that there are some real crucial contributions of these subregions to cognition. So now what happens if you were to lesion the anterior cingulate cortex? Well, it does not impair error correction or conflict-induced behavior adjustment. If you would kind of think what the conflict monetary theory is kind of produced, but then they do impair animals' ability to make optimal decisions about actions. So when you kind of think about what the intercingulate cortex is doing, it's really kind of calculating what are the most optimal decisions and kind of also think about what are the possible consequences there can be about something, some action you're about to partake. Okay, so now as the animals become deficient in updating the action values on the basis of positive and negative reinforcement histories, then basically you see that this area plays an important role when it comes to the evaluation of the possible repercussions when it comes to a certain actions you or the animal is going to take. Now, there are some electrophysiology studies that's done in vivo. So they did some single unit recording, and then so they have kind of shown that many anterior cingulate neurons. Anterior cingulate neurons basically code and encode action reward combinations. Okay, so what does that mean? Well, I mean that is basically consistent with a role of the ACC in goal-based action selection. Okay, so the encoding can also kind of represent the direction and amount of error in actions value predictions. So, I mean, which is consistent with the role that the ACC in adjusting to actions for better gain control. And now what happens? That would mean that that the lesion animals with anterior cingulate cortex lesion, by contrast, will kind of impair the animal's ability to attach values to these kind of rewarding stimuli. Okay, so that would mean that ACC really plays a very important role when it comes to the evaluation of what is to gain or to lose within a, it doesn't have to be a social hierarchy, or it can be something that is more also sort of primal when it comes to the instincts. So, uh, so basically, it's kind of evaluates what are the possible repercussions after an action has been taken. Okay, so what are some of the ACC's known projection and proposed functions? And as we said in the previous slides, the ventral ACC is connected to striatum, orbitofrontal cortex, the medial temporal lobe, and these are proposed to play a more role in emotional aspect as opposed to the dorsal ACC. It has reciprocal connections to the dorsolateral prefrontal cortex, and which is going to be more cognitive aspect of the ACC. Now, there are two kind of competing theories when it comes to what does the ACC do. So now, according to the conflict monitoring theory that you see on the left, the anterior cingulate cortex monitors or detects the presence of conflict by receiving task relevant and task irrelevant information from all these separate neural pathways. Okay, and then what happens is that kind of conveys the conveying the conflict related information to areas such as the dorsolateral prefrontal cortex, which then adjusts the level of cognitive control accordingly. Okay, now the conflict may arise at two levels of processing that you see. And then, so now let's think about this. In the stimulus or the sensory level, that's one level. Okay, when two sensory features that is either task relevant or task irrelevant of the stimulus that are processed. Okay, or it could be happening at the response level when two behavior responses that are task relevant or task irrelevant compete to gain control over the ensuing behavior. And then the dorsolateral prefrontal cortex in turn could stimulate the task relevant neural pathway by enhancing the processing of task related stimuli features and or by facilitating the selection and execution of the task related response. Okay, now then what happens now in parallel, the processing in task irrelevant pathway and task relevant responses may be inhibited. Okay, so that's the conflict monitoring theory of the ACC function. Now, in contrast, according to the ACC regulatory theory, the ACC is a part of the neural circuitry that exerts executive control by selectively biasing processing in favor of task relevant information in stimulus in situation in which there is like a need for executive control. So like all the red lines are that with the plus are basically kind of that the lines that kind of indicates the facilitation in the inhibition. So So you can see that the plus is going to be the enhancing ones, and then the one with the blunt at the end is going to be the inhibition one. Okay, so you can see that over here on the left, what the ACC really is doing is really detecting all the conflicts, and what happened when it comes to the cognitive control, then all the job is being done by the dorsolateral prefrontal cortex. Okay, and then this can happen at two levels. It can happen at the stimulus level, both task relevant or irrelevant, or it can happen at the conflict level down here, and that can be also task relevant or task irrelevant. Now, the regulatory theory of the ACC functions is basically there are two parallel circuits that's going on. So you got task irrelevant and you got task irrelevant, and then when it gets to the information processing level, then and from the information processing level to the task relevant response or the task irrelevant response, then this is where the ACC's job is doing. The ACC really is either inhibiting that or enhancing the relevance or the irrelevance of the task when it comes to these aspects. Now, the opposing theory, which is the ACC regulatory theory, that proposes that the ACC is part of the neural circuitry that actually exerts executive control as well, in addition to the dorsolateral prefrontal cortex. And as you saw in the previous two slides, it's the job of the ACC, the anterocingulate cortex, that is doing the monitoring, not the dorsolateral prefrontal cortex. Now, there is a task that is very sensitive to basically tell you whether or not if this person's prefrontal cortex, especially when it comes to monitoring of these cognitive control, whether or not if there are some sort of damage to the anterocingulate cortex, and that is the Wisconsin-Carr test. Okay, now before I kind of tell you more about what the Wisconsin-Carr test, let me kind of tell you how the test actually works. Okay, so what happened is that you start with three kind of decks. Okay, so these are the things that you can kind of match them to, and then the patient or the subject actually has a deck of cards that's in front of them. Okay, and then the task for the subjects is to sort. Basically sort. Now you'd be like, how do they sort? Well, the way that it's not being told to the person. Okay, so you kind of just say that your job is to sort them, and my job is to tell you if you're correct or if you're incorrect. So you can see that the first way, the subjects they might do is, okay, I can sort to the color of the thing, or I can sort to the number of the thing, or I can sort to the shape of the things, right? So this example here, if a person's with this card, then let's say with the 4 stars, and he is putting this right in front of the is sorting with the red triangle, then he's sorting it with color, okay? And then, so let's say if the first trial is to sort by color, and then what then what the experimenter, what the doctor is going to tell you that this is correct, this is correct, this is correct, this is correct. And then what, unbeknownst to the subject, then what happened is that after 10 consecutive correct trials, then the rule is going to switch. It's no longer going to be uh, focusing on colors or sorting by color. It's going to change to something else. It might be changing to sort by the number of the items, or it might be sorting to shape. Okay, so you can see that the person can sort by color, they can sort by number, or they can sort by shape. Okay, and then what happened is that uh, after 10 consecutive trials of, let's say, sorting by color, and then the rule is going to change, and now it's going to change to sorting by number, and then after another 10 trials, then the rule is going to change, and it's going to change to shape. And then what is going to happen is that uh, the experimenter is not going to tell the test taker, the subject, or the patient that there's going to be a rule change. So the whole point is to see whether or not the test taker can really get a feedback from the experimenter and adjust their behavior, or adjust their strategy in this case. So, and you can see that what does that mean? Well, that means that a person with proper mental flexibility, or not just mental flexibility, but also have a very strong evaluation or to the feedback, then he or she can quickly adjust his or her strategy when it comes to the sorting. Okay. Now, there are patients, especially the ones with a significant amount of prefrontal cortex damage, that they would not change any strategy. And then it's kind of like after 10 consecutive correct feedback, then they're just gonna keep sorting to color, color, color. They're gonna keep sorting to color, color. And despite the fact that you keep telling them that's incorrect, that's incorrect, that's incorrect, they don't care. They just keep going. So this means that there are a lot of cognitive inflexibility or cognitive rigidity that is going on, first of all. And the second of all is kind of like there is something called a perseveration, which means that you keep doing the wrong thing despite the fact that uh, the person who is telling you, who is grading you in this case, that's the test grader, right? The test administrator, and it keep telling you that you are wrong, that is incorrect, that is incorrect. They just keep doing exactly the wrong thing over and over and over and over again. So this just further exhibit the fact that the anterior cingulate cortex plays a very important role when it comes to two things: cognitive flexibility, and the second thing is evaluation of the outcome of a certain particular uh, motor actions, so some sort of actions. Okay, in this case. Okay, so the clinical Wisconsin card sorting exam kind of assess the ability of the participant to shift between different rules, right? This is something that we talked about in the previous slides. Now, in the monkeys, there are also a Wisconsin card sorting exam. Okay, so in this case, they are only going to give the monkey two rules to match it on. They can either match it on color or can match it on shape. Okay, now in each of the Wisconsin card sorting analog for the monkey, the sample first appeared and then three items are added. Okay, so to kind of see the figure that's in here. And now in the limited period, the monkey was required to touch the test item that matched the sample in the relevant dimension, either in color or in shape. Now, the relevant dimension was not cued and changed without notice whenever performance reached 85% correct in 20 consecutive trials. Now, the trials with two different levels of conflict, high and low, were randomly mixed. So it's not going to be all high conflict or all low conflict. Okay. And then what happened is that in high conflict and high conflict conditions, the sample matched one test item only in color and another only in shape. Therefore, the monkey had to resolve the competition between two potential responses. Now, in the low conflict condition, the sample matched one test item in both color and shape, and it did not match the other two set test items in either color or shape. You guys can see that in the response, right? So that means that there is no conflict between the matching rules when it comes to the low conflict conditions, okay? Now in the figure, the circle indicates the test item selected by the monkey. Okay, so you can see that the monkey rarely selected the item that did not match the sample in either the color or shape. Now, in the low conflict condition, one of the test items was identical to the sample, and it may be assumed that making the correct response was based on the identity of the object rather than on implementing color or shape matching rules. Now, however, the overall structure of the Wisconsin car sorting test analog did not allow this strategy. There was no cue for the currently relevant rule, and therefore the monkey had to maintain the relevant rule across trials in working memory to respond correctly in high conflict trials. So you can see that the essential rule of keeping a rule in working memory was further supported by the monkey's quick recovery of performance after a rule change. And so with not that many errors, okay? And then so one of the things is that this working memory is very sensitive to interruption. So for example, if you insert something, let's say a face discrimination trial between the consecutive Wisconsin car sorting trials, this would reduce the monkey's performance down to chance levels. Okay, so what happened is that if the monkey used object identity rather than the rule-based strategy in a low conflict trial, then working memory of the relevant rule would be compromised and performance would drop in the next high conflict trial. So that means that the high performance in the high conflict trials that were preceded by low conflict trials suggest that the monkey used color or shape matching rules in low conflict trials as well. Okay. Now, furthermore, the rule-dependent activities of the dorsolateral prefrontal cortex cells were maintained in low conflict trials and were as strong as the ones in the high conflict trials. That kind of suggests that the monkey used the color or the shape matching strategy even in the low conflict trial just as the same as in the high conflict trials. Okay. Okay, now these are the actual data from the monkey's performance. Okay, so the monkey performed the analogous of basically of the Wisconsin car sorting test, which they have to kind of match the sample to one of the tests in either by color or by shape, right? And that so the trials were either in high or low conflict, as we said in the previous slide. Now so A is showing you the mean difference in normalized speed. Okay, so this is the reaction time of the target selection. Okay, and in the low and the high conflict trials in monkeys with lesions in the dorsolateral prefrontal cortex or I mean not but or the anterior cingulate cortex. Okay, the control is in the gold color and the lesions in the dorsolateral prefrontal cortex and lesion in ACC is in the blue. Okay, and then in monkeys without a brain region, a brain lesion that which is like in the controls. Okay, now in all three groups, the reaction time was lower than in the high conflict conditions, indicating that there is some sort of adverse effect of conflict on monkeys' behavior. Okay, now B is the mean differences the in basically like this reaction time with respect to the second trial of high conflict versus so high conflict versus the low high conflict trial sequences. So basically this is a high followed by low high trial sequence. Okay, and then so now we can see that in the control and the ACC lesion groups, not in the dorsolateral prefrontal lesion group, the 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 STS was higher in the second trial of the HH pairing, so high conflict followed by high conflict. Now this indicates that the monkey were faster in resolving the conflict if they had experienced a high level of conflict in the preceding trial. Okay, so this is consistent to what I show you in the human data when it comes to the face to the name congruence versus incongruent and task. Okay, now what C is showing you is that the activity of the dorsolateral prefrontal cells represents a level of the conflict experienced in the previous trial. So you can see that this is looking at LL versus sorry, this is looking at HH versus LH. Okay, so high conflict versus low high conflict. Okay, and so this is basically, well, what is this really showing us? Well, this is showing us that let me think about it. Okay, so this is basically showing us that there is some sort of conflict when it basically showing that the high conflict trial that occurred before or after the high conflict or low conflict trials. Okay, now what happened is that you can see that the start of the cue and the fixation of the cue in the low high conflict versus the high conflict, there is more cognitive capacity that is needed during a low high conflict, but not so much with a high conflict. Okay, so that means that the task is more taxing when it comes to the low conflict followed by a high conflict sort of trials. Okay. Okay, now in B, what they're showing you is the activity of the dorsolateral prefrontal cell kind of represents currently experienced conflict. So this is what they're doing. They would be recording in the dorsolateral prefrontal cortex while the monkey is performing these tasks. Okay, now in this cell, what you're seeing is that the activity was significantly higher in the low conflict conditions. Okay, however, the cell with higher activity in the high conflict conditions were also found in the dorsolateral prefrontal cortex. Okay, so now, when we look at all these spikes, the histogram on the right, what we're basically seeing is that the black is is the low conflict, and the red is the high conflict trials, in which the same matching rules have to be applied, and which had the same correct response direction. Okay, now the stimuli that were presented in each conflict condition are shown above the individual histograms. Okay, now in the parts of C and D, the mean activities are aligned at the time of the sample trial. So, well, okay, you're probably thinking that, well, what does that really mean? So when you look at the data in these cells, you can see that the activities of the low conflict trials seem to be a little bit better compared to the activities that is going on in the high conflict trials. Okay, so that would actually mean that for the activities that is going on in dorsolateral prefrontal cortex when it comes to the low conflict trials, it's more uh, the cell fires more easily, okay, which kind of signify that there are more tasks, it's more task relevance when it comes to the easier low conflict task. Now, the same exact cell does not fire so much when it comes to the high conflict, which kind of says that either it is being controlled by somewhere else, or these cells are not that sensitive to it, and therefore make this cells a lot harder when it comes to dealing with the task that is high in conflict. Okay, so what have we learned from the previous slides? Well, it basically kind of says that this model is an extension of the conflict monitoring model with additional to the mnemonic compartment. Okay, basically it's like the short-term memory of experienced conflict in which the information regarding conflict may be held during intertrial intervals. Okay, so the blue boxes show the three main stages in conflict-induced behavior adjustment. And this model proposes that the conflict is detected is it is encoded at task relevant variable and maintained in short-term memory within the across and within and across trials. Okay, so this information about level of recently experienced conflict is then used in the subsequent trial to adjust the amount of control that is needed to enhance processing and task relevant pathways to better resolve conflict and consequently improve performance when the subject confronts similar conflicting circumstances again. Okay, now the original conflict monitoring model assumes a crucial role for the anterior cingulate cortex and the dorsolateral prefrontal cortex in conflict detection and executive control adjustment respectively. So that means the anterior cingulate cortex is kind of more important for the conflict detection, and the dorsolateral prefrontal cortex is more important for the executive control adjustment. Okay. Now, however, in the light of the recent findings, what we pretty much are suggesting is that the anterior cingulate cortex does not have a causal or this an indispensable role in conflict detection, but the dorsolateral prefrontal cortex encodes the current level of conflict maintains conflict information within and across trials and implementing cognitive control. Okay, so you can see that what this model is basically saying is that when there are in what is going on in the current trial is that there are going to be conflict detection, and then there are representative so the first thing what you have is that the representation of stimulus of the features, behavior rules, or their associated responses, and then there are going to be task relevant or task irrelevant information that's coming in, and then you at this current trial you need to detect that conflict. And now between each trials, or sometimes even within the trials, there are going to be a short-term memory or a working memory, a memory of this experienced conflict. And in the following trial, what then you need to do is to kind of remember an executive control adjustment to what you remember in this previous trials. Okay, and then what you can do is then adjust based on the sensory and the motor areas. They can kind of have an enhanced enhancing the task relevant processing pathway, and then from there you can have an improved performance. And you do this from this to the next trial, depending on what kind of uh, depending on what kind of feedback that you are getting, and also what kind of the and also based on what kind of work that you are doing, okay, and and also what kind of reward you're getting, basically. All right, so now when you're looking at this thing, you're thinking that oh not the delay now match to sample task again. Well, So in this case, this is not, these are just two variants that are coming out from the original delay non-match to sample task. Okay, in in A, what they're showing you is that this is the working memory task when it comes to the location. Okay, so they are going to show the monkey where the food is, and then they're going to let this screen down that is opaque to block the monkey's view, and then the monkey has to respond to say, to kind of see if he or she can remember where the food was underneath. Okay, so that's a working memory task. And so this one that you're seeing does not have a delay. Okay, in the delay non-match to sample task, the food is actually going to occur in the opposite side. Okay, so the one that is being shown here, this is more a pure working memory task. And then the other one is the associative memory task. So this is happening in B. Okay, and then so what basically what this is doing is that the correct food will always be underneath this thing. Okay, and it's never going to be underneath this sign. And then so they're going to have a delay, and then the monkey's task is to remember to pick the one that is associated with the food pellet. And this is what the prefrontal neurons basically are showing in terms with the sustained activity during the delay response task. Okay, you can see that during the cue, you don't see that much activities going on in these neurons going, okay? But you can see that when the neurons are, when there is when the screen is down, you can see that there are a lot of activity that's going on during this delay part. And then to the point that the go stimulus go, and then they will stop firing. Okay, what does that mean? Uh, basically means? Well, that basically means that the cell does not respond during the cue, okay? And then second thing is that the activity increase when the cue is turned off and persisted until its response. That means that during this delay response when that opaque screen is down where the monkey cannot see, the monkey is working hard in kind of remembering uh, where it's going to be or what the rule is going to be. Okay, so that is basically kind of tells you about uh, how the memory when it comes to the prefrontal cortex works. Now, in addition to the where and what information in the MT and IT cortices, in the prefrontal cortex, there are direct extension that is coming from both of these what and where information. Okay. Now, in the prefrontal cortex of the macaque, if you were to record them, and so this is the sequence of the events in the single trials, and so basically they see the fixation, and then they follow by the sample, which is in the case of Bell, and then there is a what delay, and then after that this comes in like the test subjects, right? And basically the monkey has to pick the correct answer in this case, and that would be the Bell. Okay, now the where delay, then basically that the sample is going to appear, and then the issue is going to be the monkey has to pick one of the locations that were corresponding with the previous fixation, well, not fixation, with previous sample that was shown. Okay, so what you can see is that uh, when the sample is coming in, you don't see that much activity that's going on. Okay, and when now in the what delayed trials, you can see that this cell is really firing a lot over here. Okay, and then the cells when there is then when the test object and basically and then when the next trial when the where delay comes in, you don't see that much activities in the cell. Now in a different cell, basically the firing. So this is the firing property of the cell that prefers to one location. So you can see that the neuron, this particular neuron, does not relate to basically the what delay, okay, but it responds a lot to the where delay, okay. So that means that the prefrontal cortex, it really seems that there are specialty cells that are receiving inputs from the where pathway, and there are cells that are receiving specific input from the what pathway, kind of similar to what you are seeing in the basically in the extrastriatal cortex, in the parietal, in the temporal lobe, I mean. So that were the monkey data. Well, what about the human data? So this is the functional the fMRI study of working memory. So in the delayed response task, there's a set of intact faces or scrambled faces that is being presented during an encoding period. And so then after the encoding period, a probe stimulus is presented and the subject indicates if the face was part of the memory task. Okay, so you can see that during the encoding phase, they're going to show you either a normal phase or some sort of a screwed up phase, and then all of these are going to be a set that is pretty much that's going on in the encoding phase. Okay, and then there is a delay phase, just like the delay non-matched sample with the delay working memory with the delayed associative memory that you saw in the previous slide. And then during the retrieval phase, then the subjects have to make a choice, basically. And these are the data from the fMRI study of working memory. And you can see that the BOLD response, the blood oxygen level differences response in the lateral prefrontal cortex rises during the encoding phase and remains high during the delay period. Okay. Now the magnitude of this effect is related to the number of phases that must be maintained in working memory. So what does that mean? So that means that when the activities that the blood oxygen level level differences signal really depends on how many do you kind of need to remember them in a set. You can see that the 1 and 2 phases is really not that much activity during that delay phase. But then when it comes to 3 phases or 4 phases, you can see that it requires a lot more blood in the prefrontal cortex. Okay, so that means that these areas are much more being active during that delay period. So now when you kind of go back and think about the monkey data, there are a lot of firing rate that's going on during that delay period. So this directly correlates that monkey data, which means that during this delay period, when you need to kind of briefly hold information in your dorsolateral prefrontal cortex, then all of these neurons are still being active. Okay, so in this case, what they're using more blood in the human fMRI study, they are using more blood, and therefore they are most likely doing a lot of firing there, as you saw in the monkey data. Now, this is the BOLD response when it comes to fMRI of working memory. So you can see that the BOLD response in the lateral prefrontal cortex and the fusiform phase area, the FFA, arises during the encoding and the retrieval period. Okay, and you can see that now during this delay period, you don't see that much FFA activity that's going on, but you see quite a bit of the prefrontal cortex activity that's kind of similar to the level during that maintained level task. Okay, so basically what that means is that the main job of the dorsolateral prefrontal cortex is really to hold something in the memory while during that delay period until there is a go signal, in this case, the start of the retrieval trials. Okay. Okay, I think we're going to conclude today's lecture here. And then in the next lecture, we're going to talk more on cognitive control and also the executive control.